Okay, so what's a DNA mixture and how do you interpret these things? So that's what we'll talk about for the next 15 minutes or so. This was the fingernail evidence. Moving quickly on from that slide. And the DNA evidence was from under the fingernails. There were two contributors. The victim, as we'll see in the data, comprised over 90%. It was his own fingernails, his own DNA. And there was a 6.7% of some unknown second contributor. There was a lot of DNA present. 1,000 picograms is a normal amount. The FBI used standard panels. And we know the victim contributor, so the computer was able to use all the available information, and after it inferred a genotype, then make a comparison. And then, so this is what happened. The FBI's match statistic used an inclusion method, which I'll go into a little explanation about, and that score was 13,000, given that there are only almost 13 million people in Pennsylvania. Prosecutor Krostic was open to other suggestions. The computer found a match statistic, 189 billion, by a method that I'll call addition, meaning that things add. And then a third expert, Dr. Robin Cotton, who used to direct the Cellmark Laboratory, which is a premier boutique lab. She's now director at Boston University Forensic Science Program, used a slightly more informative method that we'll get to after the break, and that method had an intermediate number of 23 million. So when you look at this, one of the first questions you may ask is, why are there different match statistics? Right? Isn't it just all the same DNA? What's going on? And if there is a difference in how it's being interpreted, how do these interpretation methods differ? And finally, as again, after the break, we'll go into a lot more detail, what results should be presented in court. So first, let me explain with a cartoon, before we look at the actual data from one of the 13 loci, what is DNA mixture data? Again, these are cartoons, and the blue is showing one contributor, say the victim. The orange is showing the two alleles, everybody has two alleles at a locus, of the second contributor. And this is indicating that there's more taller bars of the two alleles of the first contributor and less of the second contributor. That is, it's an imbalanced mixture with more of contributor A contributing their biological cells to the sample than individual B. And I'm also using the size to indicate that the alleles are different. Right? Remember, these STRs are a size-based assay, so this is smaller, then larger, larger, and larger. So what happens when you combine the cells and the DNA together from the sample, when you break open the cells in the lab to extract it, when it's amplified a millionfold, the amount that you get is roughly proportional to what came in. And that's incredibly important. It's not random. If you have twice as many alleles going in terms of the quantity of DNA, you'll have roughly twice the amount coming out. And similarly, when you detect it with a fluorescence on a DNA sequencer that reads out the signals, again, it's completely proportional to the amount of signal that goes in. And that's the basis of DNA mixture interpretation. When you use quantitative data and use the peak heights, you can determine what the ratios are. And this is done in various ways by probably most labs in the country. So let's take a look at some actual data. This is locus D7. That means it's on chromosome 7. It's a location. And this is just an indication of where it might be, the S820. You see two huge peaks. Who do you think they came from? The victim, because the victim's 93%, 93.3%. And then there's some other stuff down there. And so the question that we're interested in is, what is the genotype that the data is telling us about that unknown second contributor? Now, this happens to be a particularly easy, by eye, sort of data to analyze. You have two huge peaks. You have two other peaks that have nothing to do with these peaks. And many analysts would look at this and say, well, if I know the victim, so you can start interpreting it. And if you knew nothing at all about DNA, what would you guess with high probability that the allele pair of the unknown second contributor would be? Louder? See, and you're in the wrong field, I guess. You could be DNA analysts. So I'm going to go through the two main methods that were used. I'll explain what Robin Cotton did as well. And I'm going to compare what quantitative mixture interpretation does using the heights of the peaks to what qualitative interpretation does that doesn't. That was the main distinction, or one of the two main distinctions going on. As we said before, DNA interpretation proceeds in two steps. You start with the data without looking at the suspect, any suspect, and you infer a genotype. If there's uncertainty, there's probability. If there isn't uncertainty, maybe you'll get 100% on one particular allele pair. So afterwards, we'll make a comparison to somebody and get a match statistic, a likelihood ratio. How do we infer a genotype? If you're going to do it mathematically, what you're required to do is to consider every possible allele pair. The likely ones, the unlikely ones, the most common ones, the highly improbable ones. Computers are very good at this. They don't mind looking at billions of possibilities. They do that. For every allele pair that you consider, there may be other parameters, like how much is there. If there's more of blue, the first contributor, than of yellow, the second contributor, that's another parameter the computer can try out. What's the exact quantity? Is it more like 100 picograms, or is it more like 1,000 picograms? So the computer tries out many, many variables, but the key ones for identity are the allele pairs. It has to consider all the possibilities, and at each point, it compares the pattern that it hypothesizes based on the allele pairs, the genotypes that it's trying out, with the data. So what happens? If it tries out and it says, well, I think that the first contributor has these two alleles and there's a lot of them, and the next two alleles belong to the next contributor and there's less of them, and that's amongst the millions of patterns it tries out, that might actually fit data and would have a high likelihood. And so that possibility ends up with a very high probability. On the other hand, if you try moving around these alleles and you're not able to explain the data, you're saying there's more here, but there's nothing there, but the data really are present and your explanation doesn't fit very well, then the low likelihood hypotheses with different allele pairs that don't work very well will give you lower likelihoods. I mean, you end up with a probability distribution with those patterns, those genotype values that better explain the data will produce patterns that better fit, and that will give a higher probability. Okay, and so the rule that we say, just because it's easier than talking about any math, is a better fit's more likely it. If it fits I guess you could say if the shoe fits, wear it, but or if the glove fits, wear it. Whatever the story is. Okay, so this is what happens. Comparisons keep getting made. Let me show you how this is done, so you can get more of a dynamic feel for it. Imagine that we're going to propose what will turn out to be something like the correct answer. Okay? We begin by assuming the victim genotype. Why would we assume that 8 and 12 are there? Because we know the victim's genotype, and he's in a pool of his own blood. He's there. Now we try out every possible other genotype. In this case, we'll try out a 10-13 as an allele pair. We're also trying out 9 and 10 and 8 and 14. Everything's being tried out. We'll try it out in a 10-to-1 ratio, but the computer would also try it out equal heights, or the opposite. It tries out everything. But if we try out this particular combination of the victim being an 8-12, about 90%, and the other genotype being a 10-13 at about 7%, then when you add them together, you get a pattern of 2 tall peaks and 2 short peaks. And when you compare that with the data, the computer is doing this quantitatively, it sees that same pattern of the two tall peaks from the victim, whose genotype has been assumed, at 8:12, and at 10:13, there are two smaller peaks from someone else. And you can imagine, as you already did when you said you guessed what the answer was, there are very few other possibilities that are going to fit this. If you have this 7% unknown, there's almost nothing else you can try out as a genotype that's going to give much probability. In fact, you don't get any of the computer tries this hundreds of thousands of times, and it agrees with you. In this particular case, when there are 4 alleles, 2 large that are known from a victim, 2 small from another person, basically man and machine would agree. And the question is, how much information do we get? Well, let's figure out what the likelihood is. If we all agree there's only one possibility for that allele pair, that's a perfect match, right? That's 100%. So in the numerator, the probability of the identification of the suspect matching the evidence, if the evidence has that genotype, and there isn't any other possibility, and it shares with the suspect, we're getting the full 100% of the identification strength on the top in the numerator. What about in the denominator? What's the chance of a coincidental match? A coincidental, what if it just happened randomly, just the probability of anybody in this room? In this case, for the allele pair 1013, that turns out to be a little under 2%, 1.7%, by the product rule, by combining the known frequencies in a Caucasian population. And so the ratio of 100% over about little under 2% is over 50. You just divide 100 by 2, you get 50. And so it's a little under 2, you get a number bigger than it. It turned out to be 58 in this case. That is the likelihood ratio, the match statistic, the degree to which you believe this is an identification as opposed to a coincidental match at this one locus, D7. And that number gets multiplied by what you find at the other loci as well. Okay? And the thing to see is that if there's a high probability of that genotype, like 100%, it's definite, you retain all the probability. Now let's see what people do. And this really is what people do in virtually all crime labs around the world. Many groups do consider the victim genotype, which I'll come back to later, but many groups don't. And this is the sort of review that was done by the FBI, which is an excellent lab, but the protocols that they follow often don't yield as much information, and let's see why. So instead of saying a better fit is more likely it, the rule in inferring the genotype instead is try out every allele pair. So there are 4 alleles in the data. Try out every allele pair that doesn't go outside that set of 4. So we'll see that on the data example in a second, but it could be the first and the third may be the unknown. Well, what about the 4th? That's okay. It's included. In this case, everything that could possibly fit that data is considered to have the same likelihood. They're listed out, all 10 possibilities, the first and the first, the first and the second. A list is made, and they're all given the same likelihood. Now, that's not my method. Okay, I mean, ask your local, ask somebody else. But that's the look I keep getting from why would you do that? Well, there are good reasons why people would do that. They're not a computer. They can't model the uncertainty. They can't mathematically. They can't work with doing tens of millions of calculations. They can't use modern computational statistics. It's only 20 years old anyway. And so for a person to maintain a certain level of certainty, what they do instead is they use cutoffs, absolute cutoffs. Modern computers don't do this. Computers can determine the certainty of each peak in the data by mathematical modeling. And they do it for lots and lots of problems. in economics, social science, gene chips, physics. It's standard science. But if a person is doing this manually by eye and pencil, a felt tip pen typically, then they're not going to be able to work out mathematically what the certainty is around every peak. And if you can't do that, you're left with a felt tip pen approach. And so what they do is the lab sets a threshold. In this case, it was 150 is what the FBI used. And they draw a line through the data. Okay? Everything that's over the line is in. Anything that was under the line is out. And what you're left with are 4 allele events, or what you believe to be allele events, that are treated as if they have the same height, and there's no quantitative information left. That's how the method works. And so they're all rendered as if they were the same. And then if you look at how many possible pairings there are, 8-8, 8-10, 8-12, 8-13, 4 of those, then 3 starting with 10, 2 starting with 12, and then a 13-13. There are 10 possibilities on a list. You just write out 8-8, 8-9, and so on. That 8-8, 8-10, you wouldn't write 8-9 because 9's not included. And then what ends up happening, since all 10 of those possibilities have equal likelihood, you diffuse the probability across all 10 allele pairs. Instead of concentrating it on one result, because you're looking at it and say, well, I think that's the only one by eye, if you follow these qualitative threshold methods, which are used by every lab in the US, then you're left with 10 possibilities, a list of 10 allele pairs, and the probability, instead of being concentrated on what a computer or your eye might think is the right one, gets diffused. The key thing here is that the victim is not being used as information, right? By eye, you know if there are those 2 tall peaks came from the victim, those came from someone else. If you don't use the victim information, if you don't use the quantitative peak heights, it's not obvious anymore. Now there are 10 possibilities of what could be included, which is why it's called an inclusion method. And the result is you lose match strength. How much match strength? Let's see. So we have to ask the question, at the suspect's genotype, what is the ratio of the degree to which we believe it's an identification divided by a coincidence? Well, the coincidence doesn't change. It's still the same 1.7%, just under 2% that we had before. But now, because you're diffusing your probability over 10 possibilities and doing some adjustments that I won't go into with Bayes' rule so that they're not exactly equal, in this case, the number ends up being under 5%. That's the probability. And when you divide that number, this is no longer 100%. When you divide that number that's under 5% by a number like 1.7%, you're not getting 58 anymore. You're getting 2.5. So that's where the weight of evidence vanished. Instead of saying, I think based on the victim, based on the quantitative peak data, there's only one possible answer, it says, oh, there could be 10. I'll be conservative. And that need for people, or that inability for people to compute like a computer and just use protocols like this loses tremendous information. Let's take a look at this one locus. This was D7. And this is the score of 58. This is the likelihood ratio at each of the 13 CODIS loci. This is the score at D7. It's a 58. A number in purple, which is the inclusion method of 2.5, is a lot lower than 58. 58's more than 20 times higher. And if you do that a few times, you begin to lose information, as we'll go into. Robin Cotton's method, by the way, also cuts off the peaks, but it does consider the victim. And if you do that and you consider the victim, now you know where to assign the correct 2 alleles. And that's how Robin Cotton's method preserves information at the 2 loci where there are 4 alleles. Now, in the end, you don't just report one locus. You again use the product rule, which is a joint genotype likelihood or probability, and you multiply the probabilities together. And by multiplying them together, as we'll see in a little more detail, that's where you get the 13,000. So a logarithm. Remember logarithm from high school or college or somewhere? All a logarithm is, for the purpose of this afternoon, is the number of zeros after the number. So the logarithm of 10,000 is 4, because there's 4 zeros. Okay? The logarithm of a million is 6, because there's 6 zeros after the 1. 1, 0, 0, 0, 0, 0, 0. It's just the number of zeros. The other word for it is also powers of 10 or orders of magnitude. These are all words that say, how big is a number? The problem when you get into DNA is that when you multiply big numbers together, you get super big numbers, and you can't share them on the same scale. If you draw something, and this number is 100 billion, now you draw 10,000, you're not going to see it. So you instead count the number of zeros. And that's what we do here. There are 4 zeros, roughly, in 10,000. There are 7 zeros in Robin Cotton's number, which is 23 million. 10 gives 1 zero and then another 6 zeros. And then 189 billion is 100 times a billion, which is 2 zeros plus 9 zeros, which is 11. So a question that you might have, but I'll quickly dispatch, is this unique to this case? And the answer is no. There's a paper that we've been working on with the New York State lab for several years. It was just accepted this morning, actually, in the Journal of Forensic Science. And what it shows is the difference between computer and human review. What you're seeing is 8 different cases, each of which were solved where no victim was known, and it was a mixture with 2 contributors. And this is so on the x-axis are the 8 cases, and on the y-axis is the information, the likelihood ratio, the match statistic, expressed as the number of zeros. So, for example, this is 15 zeros, or a quadrillion. And the average is 13 zeros, or 10 trillion, over the cases. A direct comparison of what was in the case folder showed that human review, using the inclusion method, as that was used in this case, drops in its average information power, the number of zeros, down to 10 to the 7th, or 10 million. And so on average, the human review, this is even without even using a victim, by the way, which would make it higher, loses 6 zeros, or a factor of a million. And the reason this is significant is there's currently a raging debate in the forensic science literature, with vitriolic articles being written by different practitioners accusing them each of all sorts of amazing things. But some groups are really believe that people should only be using methods that throw out information. Others think they have better methods, that including computers that preserve information. And this actually, I was interrogated a panel in Sydney, Australia last month, and the people at the exact opposite end of the spectrum, the former FBI lab DNA director who believes in the orange methods, and the New Zealand likelihood ratio people who believe in the blue methods, because people can do pretty well. And the truth is they just have different goals. Because if you speak with them, you'll see the goal here is for people to be very comfortable and safe with what they're doing. The goal here, in some sense, is to keep citizens comfortable and safe in their homes, so that people are not coming in and killing them. At any rate, we'll be talking more about how all this works in the Foley case with the evidence. And then the question is what was introduced in the Foley case, and we'll reconstruct what was introduced from the trial transcript in the same order it was introduced and how it was done. Okay, thank you.